Welcome to our Integrated Pest Management Academic Seminar Series, which is designed to increase awareness of new research and techniques that advance IPM and its adoption in all types of pest management settings. These seminars are once a month during the academic year, and you can find the upcoming seminars as well as our other events like our What's Bugging You First Friday series on our website, which I'll link to in the chat. And now it's my pleasure to introduce Melissa Prusinski, a senior research scientist, laboratory supervisor, and deputy director of the Vector Surveillance Unit with the New York State Department of Health. Over the past 25 years, her research interests have focused primarily on the epidemiology, vector biology, and landscape ecology of diseases transmitted by ticks, mosquitoes, and other arthropods. She serves as Chief Coordinator of the New York State Department of Health Statewide Tick-borne Pathogen Surveillance Initiative and was the head of the former New York State Department of Health Tick Identification Service. Her recent research efforts have been the first to document some of the spread of the recent tick and mosquito-borne diseases in New York. So thank you, Melissa.
Thanks so much, Brian, for that intro, and to you and your colleagues at the New York State Integrated Pest Management Program for inviting me to provide a state of the state, per se, on ticks and tick-borne diseases and some of the research that's going on here at the New York State Department of Health. First, I'll provide an overview of the basic tick biology and updates on the tick-borne diseases known to occur in New York. And then I'll discuss our tick field surveillance work, current research on emerging tick-borne pathogens and tick-associated conditions. And I'll end with some of our public education initiatives. And hopefully my slides will advance. There we go. Apologies for the busy slide here, but we are honestly a busy work group. I work in the Vector Surveillance Unit here at the New York State Department of Health. We spend a lot of our time in the field collecting primarily ticks, but also mosquitoes using standardized techniques in support of our vector and vector-borne pathogen surveillance efforts, as well as some federally funded research on vector-borne pathogens. We work closely with our colleagues in epidemiology and the diagnostic labs at the New York State Department of Health Wadsworth Center to monitor trends in reported human vector-borne disease cases, track changes in vector distribution and density, monitor for the emergence of new vector species, and perform pathogen surveillance. We also conduct population genetics studies of vectors and pathogens. We do some forecasting work and risk analyses, as well as conduct investigations with field sampling in response to human cases of rare or emerging vector-borne disease. So why do we need to be more aware of tick-borne diseases? Well, the number of tick-borne disease cases reported in the U.S. more than doubled from 2004 to 2016. During that time frame, there were 7 new pathogens that were discovered or recognized as causing illness in people. And from 2016 through 2024, our case numbers have continued to climb nationally. Why is this happening? Well, there's expansion of areas where ticks are found, more suitable habitat due to weather and changes in climate. There's more pathogens that are being discovered, and then you have more people at risk, travelers into areas that aren't aware of the risks that tick pose, and development of previous wildland areas into residential housing. So some of you may be familiar that the life cycle of exodid or hard-bodied ticks generally lasts 2 to 3 years. And during this time, they go through 4 life stages: the egg, larva, nymph, and adult. After the eggs hatch, and that's in the spring and summer of year 0 in the diagram, the larval ticks take a single blood meal that summer. Typically, it's from a small mammal or bird, some of which may harbor pathogens. And then they develop into nymphs, or the teenager stage that you see here. Teenagers are always trouble. They overwinter, and then they emerge again in the spring of year 1 and look for another single blood meal from any variety of vertebrate hosts. And there's some examples pictured there. They may transmit or acquire pathogens at this stage. And then if they're successful in finding a blood meal, they then develop into adults and emerge in the fall of year 1. To feed again on deer, other large mammals, and pretty much anything else they come in contact with. If they're unsuccessful in finding a host and feeding, they'll then overwinter and emerge in the spring of what they say year 2 is here in the diagram for a last-ditch effort to obtain a blood meal. If they feed successfully and mate, then the female drops off the host to lay another 3,000 eggs, and the whole cycle starts all over again. And because of the 2 to 3 year life cycle, you have sort of overlapping cohorts you see in this graph in the top right, or generations of ticks. And that means that infected ticks might feed alongside naive larval ticks on the same animals, perpetuating the pathogen. And I think that shows that overlap there. Some ticks are highly host specific, meaning that they feed only on one type or species of animal. For example, Amblyomma tuberculatum, the gopher tortoise tick, feeds almost exclusively on gopher tortoises that live in the panhandle of Florida. But then there are other ticks like the black-legged tick, Ixodes scapularis, which is also known by the deer tick, that feed sort of indiscriminately on mammals, birds, reptiles, and amphibians. And because the tick needs a new host at each active life stage, and it feeds on a wide variety of species, it's a great candidate for transmitting pathogens between vertebrate host species, and that includes to humans, and we refer to this as a bridge vector. Some of you may be aware that ticks use Haller's organ. It's a sensory structure on the front legs that helps them to detect host cues, as well as infrared light. Most ticks wait for potential hosts to walk by with their forelegs sort of outstretched, and we call this questing behavior. And they then quickly grab hold of that host as they brush by using Velcro-like tarsal claws that are at the end of their leg segments to quickly climb aboard the host. There are over 800 known species of ticks, and their distribution is virtually worldwide. We have about 30 species of ticks in New York State, 10 of which frequently bite people, and 4 of these species are considered medically significant vectors. And they're shown here as adult females. You see the black-legged tick, Ixodes scapularis, the American dog tick, Dermacentor variabilis, the Lone Star tick, Amblyomma americanum, and the woodchuck tick, Ixodes cookii, which my spell check always wants to change back to cookie, but I have to be insistent here because it is cookii. They transmit a variety of bacteria, protozoa, and viruses causing various diseases here in New York. The symptoms of tick-borne illness are often generalized and flu-like in nature, so think fever, headache, fatigue, muscle and joint pain, sometimes rash. The rash can vary depending on the disease. Here we see the classic bullseye or erythema migrans rash that's associated with Lyme disease. You'll see it in the bottom image there. And the macular papular rash of Rocky Mountain spotted fever in the top picture. We emphasize to medical providers that patients presenting with generalized flu-like symptoms during the spring and summer months with a history of spending time outdoors might have a tick-borne illness. But ticks are active year-round in some regions of New York, like Long Island sometimes, and the threat of tick-borne disease is ever present. The emergence of COVID-19 has also really complicated things. There is substantial overlap in early symptoms between many tick-borne illnesses and SARS-CoV-2 infection. And unlike flu, which is fairly seasonal in New York with peaks during the colder months, COVID infection can occur at any time of the year. We've had some substantial amounts of activity in the summer, which definitely can delay tick-borne disease diagnoses. So tick-borne disease is obviously a significant public health burden in New York. Most folks know someone who's been affected or have experienced a tick-borne disease themselves. This table lists the most common tick-borne diseases that occur in New York and the number of cases reported to the New York State Health Department from 2001 to 2022. 2022 is the most recent year of finalized communicable disease data that we have. And these data are publicly available on our New York State Department of Health website, aggregated at the county level. The causative agent of each disease is shown in parentheses, and they're listed in descending order of number of reported cases here. So Lyme disease is by far the most common, with over 130,000 cases reported. It's transmitted by black-legged ticks. In fact, 5 of the 8 diseases listed are transmitted by black-legged ticks. I indicated these in the table with a little image of Ixodes scapularis next to the number of reported cases. All of these diseases, with the exception of tularemia and Rocky Mountain spotted fever, have recently emerged in New York over the past 40 to 50 years, and our case numbers have increased significantly in recent years. We see about 10,000 cases of Lyme disease each year, 1,200 cases of anaplasmosis, between 600 and 1,000 cases of babesiosis, several hundred cases of ehrlichiosis, about 25 cases of Rocky Mountain spotted fever, most of which are travel acquired, but some of which are locally acquired in New York, and between 1 to 5 cases of Powassan encephalitis and 1 to 3 cases of tularemia each year. Hard tick relapsing fever, which is an infection caused by Borrelia miyamotoi, sort of a distant cousin of the bacteria that causes Lyme disease. And cases of Heartland and Bourbon virus infection have also been detected sporadically in New York in recent years. That relapsing fever is actually not a reportable condition under New York State public health law. So medical providers and clinical labs with positive patients aren't required to report results or cases to the New York State Department of Health. So our numbers are likely vastly underrepresented here. We're working to add this disease to the list of reportable conditions, but amending public health law is a slow process, as you probably can appreciate. And though not a disease per se, red meat allergy, which is also called alpha-gal syndrome, has been associated with bites from Lone Star ticks and is something we're watching carefully as the range of this tick expands in New York. Suffolk County, New York, currently reports more cases of alpha-gal than any other county in the nation. Everyone's most familiar with Lyme disease, and if we look at this map of the historic distribution of Lyme disease cases in New York from 2001 to 2013, we see that back then, the vast majority of cases occurred in the counties bordering the Hudson River, the darker areas of the map. Black-legged ticks were abundant and widespread in this region, while their populations were more focal and patchy in the other areas of New York, though still increasing. And while the denominator data may change, the distribution of many of our other tick-borne diseases in New York is similar to the map that's shown, and it's tied to vector population density and distribution, as was demonstrated in 2 of our studies. In 2008, the New York State Department of Health launched a statewide tick-borne pathogen surveillance initiative and began monitoring black-legged ticks across New York and testing them for pathogens. We focus on Ixodes scapularis because they're responsible for over 95% of the tick-borne diseases in New York. They're sort of our, public enemy #1. But we do collect other tick species like dog ticks and Lone Star ticks to test them for their associated pathogens. This initiative was an expansion of our regional surveillance that began in 2002. As a department, we've conducted active tick surveillance since the mid-1970s, and that started on Long Island in response to an outbreak of Rocky Mountain spotted fever. But we focused on Ixodes scapularis beginning in the early 2000s, and our surveys involve standardized flagging, dragging, and walking surveys conducted on public lands during periods of peak nymphal and adult tick activity in New York. Our aim is to sort of get ahead of the expanding tick-borne disease front to identify and sample established populations in areas where tick-borne diseases are less commonly reported, while continuing to monitor changes in tick populations in our historically endemic regions. So some locations are sampled annually and some are rotationally every 2 to 5 years, but we've sampled every county since the program began, just not every year. We collect up to 50 ticks per developmental stage and test them per location individually. And we've tested over 100,000 ticks since the initiation of the program, with an additional almost 200,000 tested in pooled or batch samples for viral agents. And then these results are shared with local county health departments through direct correspondence with public and environmental health directors and vector staff, if applicable. Not all counties have vector staff or environmental health staff. Also with medical providers through electronic notifications and alerts on existing provider networks, and with the public through press releases, interactive risk maps, and publicly available data sets on our website. This map shows the locations where active tick surveillance was conducted for the years 2018 through 2023. There were 426 different locations that were sampled for ticks and over 2,500 collection events by our staff and surveillance collaborators during this timeframe. Since surveillance began, a total of 854 locations across all 57 counties in upstate New York have been sampled. This excludes New York City because their Department of Health and Mental Hygiene conducts their own tick surveillance and testing in the 5 boroughs. Many of our sites have been sampled each year for nymph and adult ticks since the program began in 2008. And we're still finalizing our 2024 data, but in 2023 alone, we visited 258 locations in 48 counties with more than 40,000 ticks collected. I wasn't lying when I said we were a busy group. So our 2023 infection rates for the most common tick-borne pathogens in nymphal, which are the immature ticks, and adult ticks are shown in the table here. Typically, the infection rate in adult ticks is roughly twice the rate for the same pathogen in nymphs from the same geographic area. So you might be wondering, what's going on here with babesia, where the statewide rate in nymphs was higher than what we saw in adults. Nymphal ticks are much more difficult to collect in large numbers than adults. They don't tend to congregate in the environment as much as adults do, so you have to cover a lot of ground. And a larger proportion of the nymphs we tested were from areas where babesia is more prevalent. So think Long Island and the lower Hudson Valley, where nymphal ticks are very high and abundant. And then where a greater proportion of the adult ticks were collected and tested from areas where Babesia microti is less prevalent. So the Adirondacks, Northern Capital District region, Central New York, and Western New York. So this just emphasizes that aggregated statewide data should be interpreted with caution, given that different surveillance locations are visited between seasons and between years. We also survey hunter-killed deer annually. This is conducted during the opening week of rifle season. We used to visit New York State Department of Environmental Conservation check stations. There's less of those these days, so now we partner with processors or wildlife butchers that prepare the deer meat for consumption. We check the head and neck of the deer for 5 minutes and place all ticks in ethanol. And then we collect a blood sample from the abdominal cavity there, and have partnered with folks down in Suffolk County DOH and the USDA to get some additional blood samples from culls that they participate in. And we record all the data from the permit tag that accompanies the animal. So why do we use white-tailed deer as sentinels for arboviral surveillance? We have 9 mosquito-borne arboviruses in New York. So West Nile, Eastern equine encephalitis, Jamestown Canyon, Cache Valley. There's a whole host of them. And 4 tick-borne arboviruses. These include Powassan, deer tick virus, Heartland, and Bourbon viruses. And their prevalence is generally low in the vector population, and their distribution can be highly focal. So white-tailed deer are excellent sentinel species because they're relatively long-lived. Arboviral infection is generally not lethal to them. They readily seroconvert, and they have relatively small home ranges. In one study, sero collected from white-tailed deer throughout New York State from 2007 to 2015 were tested by plaque reduction neutralization test at Wadsworth to detect antibodies to 9 different mosquito-borne arboviruses, and 76%, or 373 of 490 samples from white-tailed deer were seropositive against at least one virus, and 39% were exposed to multiple viruses over their lifetime. And that seroprevalence varied across New York State depending on the virus in question. I'm going to switch to one tick-borne virus in particular, Powassan virus, which was named for the town of Powassan, Ontario, where unfortunately the first recognized human case, a 5-year-old boy, died from infection with the virus in 1958. It's historically one of the least common causes of arboviral encephalitis in the US and Canada, ranking behind La Crosse, St. Louis, and Eastern and Western equine encephalitis. There have been less than 100 cases that have been reported since 1958. However, the virus has one of the highest case fatality rates among arboviruses of 10 to 15% fatality on average, and up to 60% fatality has been observed. And because infection causes severe encephalitis in humans, it's also associated with a very high incidence of lasting neurologic impairment or sequelae. 50% of people who survive Powassan encephalitis have lasting lifetime impairment. The virus circulates in an enzootic cycle of transmission among various wild animal reservoirs, and it's transmitted by ticks. And the vertebrate reservoir hosts, where people accidentally interrupt the cycle, where the ticks and the hosts and the virus coexist, that's when transmission takes place. But importantly, the virus was shown to transmit in 15 minutes or less of tick attachment. So avoidance of tick bites entirely and not just daily tick checks is required to prevent transmission. So from 1975 to 2000, only 9 cases of Powassan encephalitis were reported to the New York State Department of Health, none of which resided or had recent travel to the Hudson Valley region. But from 2001 to 2014, we had a total of 19 cases that were reported, and they're shown on the map here from a 2013 study co-authored by our group, with 79% occurring in the lower Hudson Valley, that's southeastern New York. And since then, we've had about 2 to 5 cases reported annually, and cases have spread northward and westward from the Hudson Valley, with more than 50 confirmed and probable cases in total since 2001. And it's important, I think, to note that we only see the worst of the worst. So we're kind of seeing the tip of the iceberg when it comes to Powassan. Of those 50 cases, 44 of them were neuroinvasive, which means they were likely hospitalized and had more severe illness. So Powassan is composed of 2 genetic lineages of virus with distinct transmission cycles. With the exception of a few human isolates, the majority of lineage 1 strains, or POWV, prototypic Powassan virus, have been isolated from Ixodes cookii ticks and their hosts, primarily woodchucks, skunks, and other canids. Lineage 2 strains have been isolated predominantly from Ixodes scapularis and white-footed mice, Peromyscus leucopus. Phylogenetic analysis of the 2 lineages determined that they shared a common ancestor 484 years ago, and that simultaneous radial divergence occurred about 200 years ago, with no indication that one clade is ancestral to the other. And while divergence of DTV, or deer tick virus, and Powassan may have occurred well before the current expansion of black-legged ticks in New York State and may have been driven by other factors, the result was that DTV was better adapted to be transmitted by Ixodes scapularis. And increasing tick populations and increasing human contact with this tick species in recent decades has been hypothesized to have led to the observed increase in Powassan encephalitis in recent years in New York and other northeastern states. And that's basically attributed to Powassan lineage 2, or deer tick virus infection. With respect to sentinel surveillance of tick-borne viruses in white-tailed deer, Powassan virus activity was observed across New York State, as you can see in the map here, but especially high in the Hudson River Valley, where 95% of the deer sampled had antibodies, and the Capital District, where 85% were positive. Heartland and Bourbon virus activity is highest on Long Island, which corresponds with the distribution of the Lone Star tick vector in New York, and those data aren't shown here, but they're described in 2 New York State Department of Health studies that are cited on this slide. This slide summarizes the corresponding tick results from that study. Over 6,000 ticks were collected from 736 deer, and there were 96 Powassan virus positive pools from 71 animals that were harvested in the counties that are shown in purple on the map. We were also able to preserve alive, fully engorged female ticks and allowed them to naturally oviposit in the laboratory. And we documented transovarial transmission of the virus from naturally infected female ticks to their progeny, which had never been done before. They've done transmission experiments in the laboratory, and we knew transovarial transmission is a factor, but we never documented it from naturally infected ticks. Recently, we collaborated with researchers across the northeastern US, including folks at Yale University, to reconstruct the dispersal history of Powassan lineages from ticks using phylogeographic analyses. So those results for year 1990 through 2020 are shown in figures A through D. So you can see the initial clusters in the coastal areas here and here are then spreading inland over time. Figure E shows the overall effective size of the viral population, which increases over time. Figure F is a graph of the number of confirmed human cases with big increases observed after 2010. And G, you can see an estimation of the white-tailed deer populations in New York State and Connecticut. H includes environmental factors that were included in some of the analyses and modeling to test their impact on Powassan virus dispersal. And basically, they revealed that Powassan virus tended not to circulate at higher elevations, but that the other variables examined could not account for the pattern of dispersal across the landscape. So there's still much to learn about the factors that govern Powassan virus dispersal and emergence. I'm going to switch gears now to anaplasmosis, our rising star with respect to tick-borne disease in New York. It's caused by the obligate intracellular bacterium Anaplasma phagocytophilum, and it's transmitted in the Northeast and Midwestern US by Ixodes scapularis, again. The pathogen transmission occurs after 12 to 24 hours of blood feeding, so it's a little faster than the transmission of Borrelia burgdorferi, the Lyme spirochete, which is generally 36 to 48 hours. Nonspecific flu-like illness, fever, fatigue, muscle aches, headaches are some of the common symptoms. Severe complications and death occur rarely, but 35% of cases are hospitalized, and it has a fatality rate of 0.5% in New York. Treatment is with antibiotics like doxycycline. The majority of anaplasmosis cases have been reported from the eastern part of the state, but our vector surveillance indicated the greatest risk was in those regions, but also indicated that there was risk in central Western New York and on Long Island. So you can see on the right here, we've got hot and cold spots as the base map. So the red areas are where we're seeing anaplasmosis. The blue areas are where we're not really seeing very many reported cases. And then the dots that you see and the circles overlaid are the entomologic risk index, or the prevalence of the pathogen multiplied by the abundance of ticks. So the larger the circle, the higher the risk, supposedly for anaplasmosis. And you'll see that in general, most of the large circles overlap with where we see that hot spot area, the red area of the base map. But there are some decent-sized circles in Western and Central New York and on Long Island where we were not seeing very many reported cases. So we were concerned, was there underreporting going on, misdiagnosis, or some other process at play? So there are multiple genetic variants of Anaplasma phagocytophilum, and they basically tend to pair up with different host species. Humans are only susceptible to infection with the human active variant, or APHA. It's A, or the red variant that you see in the slide image here on the right. Major reservoirs of this variant in nature include white-footed mice and chipmunks, probably some other small mammals. But there's also a non-pathogenic, or AP variant 1, which is B, or the blue one in this slide. And that one is primarily associated with white-tailed deer and doesn't cause clinical illness in people. It's important to note that the ticks are infected during blood feeding. The infection is maintained through the molt from larvae to nymph to adult transstadially, but there is no transovarial transmission. And anaplasma infected ticks, interestingly, are more cold tolerant, so they may be able to better overwinter and remain active at lower temperatures. This was documented in a laboratory study, but it'd be interesting to see if we see a higher proportion of anaplasma infected ticks survive to the next spring. Our tick surveillance pathogen testing doesn't differentiate between the pathogenic and non-pathogenic variants of anaplasma. So the initial test, which is a quadplex assay that looks at the bacteria that causes Lyme, anaplasma, the pathogen that causes babesiosis, and then that relapsing fever spirochete, Borrelia miyamotoi. That quadplex doesn't tell us whether the Anaplasma phagocytophilum is the human active variant or the non-pathogenic variant. We run a second test called a single nucleotide polymorphism, or SNP assay, to differentiate our anaplasma positive samples as either the human active variant or the deer variant. The allelic discrimination plot on the bottom right shows what the results look like. So you see the green circles are the human active variant, the blue circles are the deer variant. The red circle that you see there is a co-infected tick sample, which is actually quite rare. And then the black square represents the controls, the negative controls for the test, where there's no template. So we saw an improved association between anaplasma HA, or human active prevalence, which is the red circles on the map that's hopefully animated for you, and the human anaplasmosis incidence, which are the red areas of the base map. And that applied over both space and time. Then when we were looking at the overall anaplasma prevalence, when we weren't differentiating. So we definitely saw improved association. The blue circles are the non-pathogenic variant that clustered in areas with fewer reported anaplasmosis cases. So those are the blue areas of the base map. And I'm going to switch gears again because I want to cover as much as possible. We're going to chat a little bit about alpha-gal syndrome. And though not a disease per se and not a reportable condition in New York State, alpha-gal syndrome is something that's very much on our radar at the New York State Department of Health due to its association with tick bites. So you've got, it's a serious, potentially life-threatening allergic condition that can occur after a tick bite. It's primarily associated with Lone Star tick bites, but other tick species may also be involved. It's named after galactose alpha-1,3 galactose, which is a molecule found in most mammals. Symptoms appear 2 to 6 hours after people eat red meat or exposed to other products that contain alpha-gal, like milk, and there's some pharmaceuticals as well. The symptoms may be mild or severe. They can range from rash or upset stomach to full-blown anaphylaxis. Alpha-gal syndrome reactions can be different from person to person, and people may not have a reaction after every time that they consume an alpha-gal containing product. From 2010 to 2022, more than 110,000 suspected cases of alpha-gal syndrome were identified in the US. These were mostly adults in the South, East, and Central states. Basically, the test involved to determine whether somebody may be a potential alpha-gal syndrome sufferer involves looking for SIgE antibody. That's key to the alpha-gal diagnosis, and that the presence of that antibody may, but doesn't always indicate that a person may have alpha-gal syndrome. There was a newly drafted national case definition, and it's intentionally very broad to make sure that we aren't missing any cases, but it will likely be refined as we learn more about alpha-gal syndrome. All persons who test positive for that alpha-gal SIgE meet the case definition for a suspected alpha-gal syndrome case. And then it's basically confirmatory laboratory evidence, but no clinical information. So a retrospective analysis was performed by CDC to understand the distribution of alpha-gal syndrome nationally from January of 2017 through the end of the year 2022. Viracor IBT Laboratories was the primary commercial lab that tested to detect SIgE antibodies to alpha-gal until August of 2021. They were the only ones. This analysis also evaluated epidemiological variables provided with the lab samples at the time of testing. So they're kind of limited. It's just basically patient age, sex, date of sample collection. But there's no clinical data available. So many of these suspect cases might not actually experience symptoms after consuming alpha-gal containing products. Again, it's likely an overestimate of our alpha-gal cases, but there's a hot spot you see up here in the Midwest, in Minnesota, in Wisconsin. It's of particular interest because Lone Star ticks are not known to be established in this region. It's also important to note that clinical suspicion and referral of patients for SIgE alpha-gal antibody testing likely varies geographically. So you might have some providers that are on the ball and they're sending folks for testing, and other places where they're less familiar and so they're not sending people for testing. And travel history was also not collected on these suspect cases. So both of those can impact interpretability of these results. But still, it's you can see the South where Lone Star ticks are widely distributed and abundant, and you definitely see the darker areas of the map there. Further analysis to understand state-specific trends in case distribution was done by CDC. There were 4,091 New York State cases during the study period, 90% of which were residents of Suffolk County. We have not detected established breeding populations of Lone Star ticks north of New York City, but nymphs and adults have been reported from nearly every upstate New York county through our passive surveillance or Tick ID service, which ran through 2021. These are likely migratory bird dispersed ticks that were moved from areas where Lone Stars are more common, but it's important to note that black-legged ticks are abundant and widespread across much of upstate New York, and their potential role in alpha-gal syndrome really hasn't been fully determined. The total number of alpha-gal SIGE tests ordered has increased over time in New York, and you can see that in the graph on the left there. And test positivity overall, which was 34.2%, is similar to the national average, which is 30 to 35%. There's also no predilection for test positivity correlated with sex, but positivity increases with age, as is shown in the graph, the red one on the right there, you can see. And this graph here shows Lone Star ticks submitted to the New York State Department of Health Tick Identification Passive Surveillance Program from New York residents with no reported travel outside of New York State in the weeks prior to tick bite. You can see that submissions increased from 1 in 1991 to 224 in 2009. And while the service was discontinued in 2011, and these data don't overlap with the data presented by CDC, which are 2017 through 2022, you can clearly see an increasing trend in Lone Star tick encounters by New York State residents, and that likely continued into the years beyond what's shown here on the graph. Locally acquired Lone Star tick submissions from Suffolk County residents in particular mirrored this statewide trend, which is why I didn't bother putting together a second chart with their submissions. It looks just like this one. So this map was created showing the incidence of people testing positive for alpha-gal SIGE antibody per 100,000 population at the zip code level in Suffolk and Nassau counties for the 2017 through 2022. And this is using that VeraCore lab data that was provided to us by the CDC. Incidence is shown along a color gradient from yellow to red, with increasing incidence observed as you move eastward across the county. This pattern is likely tied to Lone Star tick population density, which is based on population estimates from active surveillance that's conducted by Suffolk County Department of Health, and the resulting tick encounter risk. Tick bites, from the species are associated with alpha-gal syndrome, but really further study is needed to identify other potential risk factors that may influence incidence of suspect alpha-gal syndrome cases in New York. So speaking of active tick surveillance, our publicly available data sets are provided here from the results of New York State active tick-borne and tick-borne pathogen surveillance, and they're aggregated at the county level. We use these to educate the public and medical providers about risks of coming in contact with ticks and tick-borne pathogens. The data arise from surveillance at precise locations at one point in time. It's important to note that and consider that. Both measures of tick population density and pathogen prevalence can vary greatly over a small geographic area and like within a county. So these data shouldn't be used to broadly predict disease risk for a county or region, but it does let you know what the rates look like and what seems to be circulating in the various counties of New York State. In addition to the work we've done to educate the public through Health Data NY, we have various media campaigns and videos that are available on both the New York State Department of Health's public website and YouTube: how to safely remove a tick, appropriate repellent use, which is a video starring my finger, and that's available in both English and Spanish language. We have some videos that detail our active surveillance and testing, as well as prevention measures, including repellent use. And we did a couple podcasts with on anaplasmosis and Heartland virus. We were invited by CDC to participate in their emerging infectious disease podcast program. We have warning signs that we try to get up on public lands, particularly if we see increasing tick populations or new pathogens emerging, tick removal kits, which we distribute at the New York State Fair, and events with scouts or hunters groups. And we collaborated with the New York State Ed Department to develop and distribute a tick-borne disease education toolkit in schools. Basically, it was advertised to nearly 4,000 school nurses, health educators, medical directors, and school administrators. And we're always looking for new ways to educate folks about ticks and tick-borne disease risk. We're considering QR code accessible tick forecasts that would be available at trailheads in parks, just-in-time tick bite prevention training. It would also give you links to our tick-borne pathogen surveillance data that are available publicly and risk maps. People would basically just scan this with their smartphone. Maybe they could opt in to receive SMS text reminders to do a tick check at certain time intervals during and after spending time outdoors. These are some things we're considering and sort of like spitballing to see a level of interest and if people would be willing to scan a QR code to get those reminders. And all of our education materials and outreach methods contain consistent messaging. And I'd be remiss if I didn't stick this prevention messaging after hearing about all of these tick-borne diseases and tick-associated conditions. We always stress to folks to wear light colors and pull up your socks over your pants. If you're going to be out there for a long time, maybe you're doing some brush clearing. I use clear packing tape and tape that juncture between my socks and my pants so that it limits the access to the skin and the ticks are forced to crawl upward. And because you're wearing those light colors, you can readily spot them and flick them off before they attach to your skin. Frequently checking, obsessively checking when you're outdoors, considering using repellents, 40% or greater DEET, 20% or higher picaridin, IR 3535. Treat your gear and your clothing with permethrin. That's not safe for use on skin, but is for treating clothing and gear. There was a study that CDC did with some folks in Rhode Island that showed if people shower within 2 hours of outdoor activities and do a full body tick check, greatly reduces their chance of getting a tick-borne illness. And of course, prompt and proper removal of ticks using fine tip tweezers only, no other method. And we have that video that shows you how to do it, where I got to attach clean nymphs to my boss's arm, and then we removed them to show you how to do that. And then lastly, a huge thanks to our public health and academic partners that help make all of this work possible. And with that, my contact information is there, and I'd be happy to stop sharing and answer any questions.
Thank you, Melissa. That was fantastic. That's a huge amount of work that your team has done there. So yeah, we have about 10 minutes for questions and comments. You can either unmute yourself to do that or write it into the chat. And actually, I see we have a couple already, which I'll read aloud. First from Nick. How many of those signs can we get? Our local St. Lawrence County Public Health Department and Cornell Cooperative Extension are attempting to put signs up at all the trailheads in the county. Currently, we're putting up the CDC sign, but our New York State Department of Health sign would be better.
Email me and I will send you the form where you can request them. They were limiting them to 5 per location or organization, but I'm sure that St. Lawrence is one of those counties where we are seeing increasing tick populations, and we would love for the signs to get out there. So I will definitely, in fact, maybe I was going to try to cut and paste a link and put it in the chat, but it might be safer for me not to do that because last time I lost my communication capability. So, but yes, please reach out, and we have a standard form you fill out. You can request any of our free educational materials. We have a stockpile of them in the warehouse.
Okay, great. And I just put your email into the chat for folks in case they
Thank
didn't
you.
copy it down earlier. Next question from Courtney. I'm doing an educational program for about 40 kids in the Ashokan watershed area in April, grades 3 to 6. How can I get some educational materials like trifold cards, and removal kits for them?
Same answer as for the signs. Email me and I'll get back to you with the order form. The tick removal kits are in more limited supply, but I can ship them out directly from my location here at the Empire State Plaza in Albany. And then the trifold cards come from our distribution warehouse, and we have lots of those. So definitely can get those to you.
Great. And here at New York State IPM, we also have some tick kits. And so hopefully Joellen or Jody can put information in the chat about how folks can get those as well. Next question from Eddie. Given the extremely cold winter, will the tick population be affected?
I wish, I wish. As long as there is snow, it provides this layer of insulation, and the ticks basically they're in the upper soil layers and in the leaf litter, and as long as there's snow, even if there's brutal cold, they really don't experience the same temperature fluctuations and relative humidity fluctuations as if there were no snowfall. That protective barrier really they survive without much issue. Where we definitely will see the effects of cold weather on them is when you're in the high peaks, if the snow is wind blown off and you've got bare mineral soil or very little snowpack, that will reduce populations because they will experience the extreme cold and low relative humidity, which would cause overwinter mortality.
Okay. Yeah, interesting. That makes sense. All right. I don't see any other questions. Feel free to unmute. Although here's one from Dana. With regard to, and how do you say it? Is it Babesia,
Babesia, yep.
the genus? We see less of it in adult ticks here in New Jersey as well. There is some mounting evidence that it isn't acquired as easily by nymphs as larvae, and can be lost in the adult molt. I
Oh,
guess.
well, excellent. I just thought it was in our data due to the fact that we test a lot of adults from emerging areas where Borrelia is very common, but Babesia is less prevalent. So that's interesting to learn that you're also seeing less of it in your adult ticks. I just thought it was because a greater proportion of our nymphs were collected from areas where Babesia was more common. Cool.
Great. Maybe one final question or comment. Oh, here's one from Joellen. With Gulf Coast tick now in New York, do you predict a higher rate of, is it rickettsia diagnosis?
It's certainly possible. We've seen the Gulf Coast tick historically, adults every year from most of New York State. They seem to be associated with meadowlarks. And so as they move around the state, they tend to rain these Gulf Coast ticks down, but they're not really established in breeding north of New York City right now. But yeah, we have seen Amblyomma maculatum. We haven't seen an increasing bump in our rickettsial positives, but with them being a vector species that's involved, obviously there's a greater chance that could emerge and become a problem, and is something we'll keep an eye on for sure.
Great. And actually, we got time for one final question, I think, from Dan Gilrain. Are you interested in receiving ticks submitted by the public from certain areas around the state to help inform your surveillance? And can you comment on the situation around New York with a longhorn tick?
Yeah, so we are not currently accepting citizen science contributions to our active tick surveillance program, mainly because The sampling protocols are quite labor intensive. It has to be over 1,000 meters sampled at each location. And we have these surveillance quotas that we're looking to attain from each location. 50 or more nymphs, 50 or more adults. In fact, 100 or more is preferable, so we have a decent sample size to send for arboviral testing as well. So because of that, and because we want folks to follow some very strict protocols, we haven't really leaned on citizen science. Now, that being said, you know, funding scapes can change, and our program may have to adapt over time to remain sustainable. But with our public health partners at the local county health departments and also some academic institutions, we are able to cover the state pretty well, as you saw in the slide with the map. But there are some services out there that will test ticks collected by the public. Just not us right now.
Okay, great. Well, thank you, Melissa.